Okay, good afternoon, everyone. We're just launched this webinar session and just giving a couple of minutes for everyone to join with us today. And we will be starting in a minute once we have most of the people connected, and we'll be happy to join you during the session. Okay, so yeah, since people is starting to join, I suggest that we start this session. So good afternoon, everyone, and welcome to this webinar entitled Image Processing Pixels to Phenotypes to Advance Legume Breeding. This activity is organized in the framework of the Horizon Europe Project BELIS. BELIS stands for Breeding European Legumes for Increased Sustainability. My name is Joaquin Balduque. I'm the coordinator of the Plant Production, Health and Breeding area within the training unit of SIAM Zaragoza in Spain. SIAM Zaragoza is the Mediterranean Agronomic Institute of Zaragoza, the Spanish Institute of SIAM, which is the International Center for Advanced Agronomic Mediterranean Studies. We are leading the work package dedicated to the the work package BELIS Breeding Actors platform, networking, communication, dissemination, and transfer. And before we start, and for your information, I would like to show you and share that we have created the Q&A section within the session, so you can go there and don't hesitate. If you look closely to define, you will find the option of the Q&A at the bottom part of your application. So if you have any questions, please type them directly, and we will address them as the most appropriate time. And I would like to thank all attendees who are joining us today, as well as all the BELIS colleagues who will be presenting during this webinar. So going directly to the opening, as you may have seen in the promotion of this event, the main objective of this session today is to showcase how high resolution UAV imagery combined with spectral, spatial, and AI concepts enables non-destructive, time-efficient extraction of quantitative phenotypic information from crops. So through different focus presentations, we will be introducing the fundamental remote sensing concepts, demonstrate the translation of pixel level data into plot level phenotypic traits, and discuss the transformative role of automatization and vision-based AI in modern plant phenotyping. The session will also present practical case studies in Lucerne, red clover, and pea, comparing drone-based predictions with manual phenotyping and evaluating the impact on working conditions and equipment investment. And furthermore, the European Project BELIS and the stakeholder networks established within the project will also be introduced. So after this opening, I will stop here and we will move to the first presentation as outlined in the program. We are going with the BELIS Project Coordinator, Dr. Bernadette Julier from INRAE France, who is going to present the project. Bernadette, thank you very much for being there, and the floor is yours.
Okay, thank you very much, Joaquin, and hi, everybody. Thanks for joining this meeting or this webinar. I share my screen. Can you see it?
Yes.
Yes, okay, thank you. So I am the project coordinator. I am a scientist working on the genetics and at INRAE near Poitiers in the unit who works on forage crops and yes, forage crops, that's it. Okay, so what about the BELIS project? Maybe some of you have already heard about it, but not all of you. So quick presentation. Sorry, I have to go to this. So the concept of the project is...
Sorry, Bernadette, you stopped sharing somehow.
Oh, I have stopped sharing. Oh, sorry.
Yeah.
What happened? Sorry.
No problem.
So Yes. Okay. So the concept of the project is to go to all the steps going from legume breeding to use of the varieties. So of course breeding that goes to improve varieties, and then the varieties goes to registration, and they are then allowed to go to the market, and you have series of evaluation and recommendations. And of course, this is possible because of cooperative models between actors of the selection. Oh, but I don't know how to go from one slide to the next one.
Oops.
Sorry. Okay. So, yes, I think it will work. Okay.
Yes.
So it's okay now. Sorry. So we have identified a number of lock-ins, different steps. First at the genetic step, then on the regulatory events and on the recommendation also, and of course there are a number of financial lock-ins with little support to legume breeding. So we will try to progress on these different aspects. So with, for example, a better exploitation of genetic resources, improve the breeding methodologies with better phenotyping, low-cost geotyping, and also go to the proof of concept in breeding. For the regulatory and recommendation lock-ins, we will improve registration process and recommendation, and we will also study have case studies and living labs to improve the cooperative models. So this work is done at the scientific level, let's say, and we have also a platform that gathers all type of actors interested in legume breeding and genetic progress. And so this platform gathers people such as breeders, extension services, registration offices, researchers, and also industries, feed and food industries. We have set up the BELIS network, that is a network dedicated to selection of legumes in the EU, of course, associated countries and beyond. And we have a portfolio for training and dissemination with online tools and databases, videos, training session, workshop, and webinars. And today is one of this webinar. So the three main objectives of BELIS are to develop tools and methodologies for cost-effective breeding programs and deliver proofs of concept with and for breeders. It's also to facilitate the economic and regulatory environment, so for variety registration, variety recommendation, and cooperative models, and also to implement an efficient, ambitious, and durable transfer of innovation. So this BELIS platform that includes the network of breeders and actors from scientific research, extension services, and food and feed industries, as well as training portfolio. So we are working on 14 legume species, 7 grain legumes and 7 forage legumes. We are not working at the same intensity on each species, but just to remind you that grain legumes is used for food. All types of legumes are used for feed, but for sure, forage legumes are not quite often used for food. They don't feed the same animals. Grain legumes mostly are used for monogastric animals, and forage legumes are used for polygastrics or ruminants. And all species provide very important ecosystem services, and for sure forage legumes produce a little bit more, but all of them are important, especially for nitrogen, related to nitrogen cycle. The partnership of BELIS is composed by research institutes on the left-hand side, education, so IMZ, CIAM, seed industry with 9 partners, extension services with 3 partners and 1 registration office. And this Daisy the earth of this daisy, of course, are breeders, and the leaves are consumers, feed and food industry, and farmers, even if they are not directly partners of the project. You have a map where are located the partners on the right-hand side. A few examples of the expected results. For example, we want to progress on the high throughput phenotyping, and this is the topic of the webinar today, with use of drone flights and imagery. And that could be implemented by partners on breeders' plots with breeders' material and all this application. But we have also other topics on this subject, with refinement or sharing of other phenotyping methods related to disease resistance, quality traits, insect resistant or drought resistant, etc. We have also a topic on high throughput genotyping, and for example, we are building a grain legume genotyping array with a multi-species interest for grain legume, and the objective is that the genotyping cost is lower than the plot cost. But we are also genotyping plant collections from genetic resources or breeding materials, and with the objective to have a better use of the genetic diversity. And we want to make use of the markers for distinctness tests in variety registration. We are developing tools for molecular breeding, and we want to revise breeding methods to speed up genetic gains. And so we want to detect, or we are currently detecting markers associated to traits, but also create or improve prediction equations for genomic selections. And we go up to proof of concepts in breeding schemes. We want to improve variety registration. And one key idea is to benefit from phenotyping capacities in EU countries. And we are testing the relevance of EU network of trials at algorithm scales instead of country member state scale. And we are using common controls across countries. We will collect all the data and imagine that each member state make his own decision. But also we are working to improve variety recommendation, and some partners are gathering data from variety value and market, and we are also modeling the adaptation of a species depending on the variety type in the EU, and to model the project performance of the species and varieties. And we are also improving breeding cooperation to support legume breeding that is invested. And so this topic is to analyze the cooperation that are possible at the moment among private breeders, public breeders, research, cooperatives, extension services, and processing industries where relevant. But we are also analyzing the effect of public policies on this cooperation. The objective is really also to communicate and disseminate results and information during the project and beyond, and with this establishment of the network to activate the flow of information and cooperation among actors. And at the end, the final objective is really to increase nitrogen fixation and protein production in Europe for sustainability and sovereignty. So I will continue my presentation by a presentation of the BELIS network. The colleague in charge of this network is Claire Barbet-Massin, but she's sorry she couldn't join today. She works at Terre Innovia in France. And so the aim of this network is to link breeders, so these actors that I've already mentioned before, in order to make available the knowledge obtained during the project, but also to facilitate exchange and collaboration among stakeholders. So it can be technical collaboration, it can be business collaboration, etc. So it's really a gate to transfer BELIS outputs and also to connect legume breeding actors across EU and associated countries. This network is also to seek for opportunities to cooperate within existing networks to avoid any overlapping activities. And there is a network advisory board that ensures the proper monitoring of the network activities and its impact on legume breeding. And so during the life of BELIS project, this network is active, but by the end of the project, it will the board will refine the network long-term aims to let it alive, to continue this network after BELIS. What are the activities of this network? First is to facilitate the access to BELIS results through different tools. First is the BELIS newsletter that regularly informs stakeholders about the project progress, new findings, and upcoming events, such as this webinar, for example. There are some surveys to refine training and webinar needs, identify thematics of interest. There are dissemination webinars to present some outcomes of the projects. We can have structured feedback sessions to gather feedback from stakeholders on the usefulness and potential application of the project, of the results. We can have thematic webinars to deliver focused webinars on specific aspects of the project results by species groups or others. And we have training activities, so in-person sessions to train stakeholders on how to use the results. The second activity is to facilitate exchange and collaboration. We can have in-person meetings to share knowledge and provide opportunity for stakeholders to meet, exchange ideas, and to identify synergies or collaboration opportunities. So recently we have had a side event of the International Legume Society conference on the 8th of, not January, but June, in Dubrovnik. And so we have this exchange. We have online meetings. So it's events with a short presentation from stakeholders, roundtables, and group discussions. We can connect with other existing networks, and of course, we are connected to Legume Generation, the sister project of BELIS, and for example, International Legume Society and others. We can also study funding opportunities from national or international EU calls or from direct collaboration among members. So the vision of BELIS is that sharing innovation, know-how, experience, and means among breeding actors is at the heart of genetic progress in legume crops. So why you would like to join? First to access the BELIS research results, of course, stay updates on the advancements, collaborate with experts, so you connect with breeders, industries, researchers across Europe and beyond, to participate in events, so webinars, training sessions, and other networking opportunities. And it's free, so membership is free throughout the duration of the project. And how to join, my last slide, I think. You can go to the BELIS website. You can fill in a membership form on this website. You fill it out, you accept the conditions. And you can also register to the BELIS newsletter. So thanks for your attention.
Thank you so much, Bernadette. Thank you for this clear presentation. And we are now moving to our next slot, which is the presentation from Arno Kasprak and Peter Lutens from ILVO. whose lectures is one of today's main topic, actually, with pixels to phenotypes, spectral, spatial, and AI concepts in UAV imagery. So thank you both very much for being there, and the floor is yours.
Okay, hello everybody. I'm Arno, and together with Peter, I will present some fundamental and basic spectral, spatial, and AI concepts in order to understand the rest of the presentations. So we will talk about in this presentation first about GIS. So what is GIS? Then Peter will talk about remote sensing for vegetation monitoring, and then also from how do we go from unmanned aerial vehicle flight to image data. And then I will end this presentation with some concepts about artificial intelligence and deep learning for remote sensing applications. But first, GIS. So GIS stands for Geographical Information System. Basically, a GIS, shown here also on the slide, is a system designed to capture, store, manipulate, analyze, manage, and present all types of geographically referenced data. The last words here of this definition are the most important one, because it's really specifically for this geographically referenced data or data that has a location somewhere on Earth. Basically, you can compare a GIS to an Excel for data, but then this time a GIS is specifically for geographically referenced data. What is then this geographically referenced data or geodata? This is data that is descriptive. So first of all, it answers to the question what. So it can, for example, tell you something about population size. It can also be dynamic, so can tell something about population size somewhere in time. And then also the most important thing is that it is spatial, so it has a location attached to it. You have two big different types of geodata that you can use within a GIS system. First, you have raster data. And rasters are basically matrices made up of pixels, as you can also see here on the slide. So you see it's just a matrix and then with different pixels, and yeah, every cell can contain a specific value. So for example, if you look at the right example here, this is an orthomosaic. This is basically an image of an entire field captured by a drone. That's raster data, because if you zoom into it, then you see it, yeah, it consists of pixels, and every pixel has a different color, which is then linked to reflectance values measured for red, green, and blue captured by a drone sensor. Very important, again, this is not just an image, it's an image with a location. An important concept for rasters is spatial resolution. So what's spatial resolution? Spatial resolution is basically it's the fixed size of every pixel in the matrix of the raster. You can have very high spatial resolution data, as here on the left, where every pixel is around 0.2 millimeters. Then you see a lot of detail. But on the other end, you can also have very low resolution data, as here on the right, where every pixel is then only 2 centimeters. It's always a trade-off between how much space do I have on my computer and how much detail do I want in my image. If you have very high resolution, then you see a lot on the image, but this requires, of course, a lot of space on your computer. So we already talked about raster data, but on the other, you also have vector data, which is another geodata type which you can use in GIS. As you can see on the slide, vector data is not anymore a matrix, but these are like simple geometrical features which can be used to represent a real world entity somewhere on the Earth. Like, for example, you can use a point vector layer. You can also use a line if you want, for example, to represent a river or a road in GIS. And you can also use polygon vector data for, yeah, if you want to represent an area, for example. Here on the next slide, I show you some examples of vector data. So what you see here is, you see here at the left, you see some GPS measurements as a point vector layer. So every point here is a feature of the vector layer. It is basically a location measured in a field, which you then can represent in a GIS environment. But what you also can do is, for example, use vector data to represent the trial plots if you have an experiment for breeding. And the nice thing about vector data is that you also can link additional information to it, like you see here on the right, where you then can also link flower counts to every plot. So here again, an overview of the different data types that you can use in GIS. So you can have a raster layer, which is always an image, so a matrix with pixels. Then on top of it, like here in the image, you see some red rectangles. These are then a vector layer, and you can always link additional information to this vector layer by using an attribute table in GIS, which looks like a normal Excel file, but here you just link additional information to every feature of the vector layer. Like for example, in this example, you link plot or class ID to every rectangle here. Okay, so we now talked about what the GIS is, what the different data types you can use in GIS, but of course we also need to know how to locate data somewhere on Earth. And for that you need to understand something about coordinate systems. And the first type of coordinate system I show you here is a geographic coordinate system, which basically uses a three-dimensional spherical surface to define locations on Earth. So imagine you have the Earth, the globe. You just draw meridians and latitudes on it, and then define an equator and a prime meridian. And then based on that, you can always define your location on Earth, as shown also here on the figure. And then, for example, we can locate Ilvo here in Merelbeke in Belgium as 51 degrees latitude and 4 degrees longitude. But it is not as simple as I just showed you, because in reality, the Earth is not a perfect sphere. So it's not a perfect globe. You have mountains also at the poles, as you can see also in the figure here. The Earth is a little bit more flat. So it's not very easy to define such a three-dimensional coordinate system. And that's why people designed mathematical representations of the Earth, so simplifications of the Earth, to make it easier to define a three-dimensional coordinate system. And such a mathematical simplification model of the Earth is called a geodetic datum. And you have different ones. You have, for example, WGS84, which is also used for a global positioning system. This is a global one, so it's really designed so that it is good for every place on Earth. But you can also have local geodetic datums that only are good to define coordinate systems at very specific locations. It's also very important to know that if you are measuring coordinates in different coordinate systems based on different geodetic datums, that they can have the same coordinates, but then different locations because you're just measuring in another reference system. Okay, but yeah, now we know how to locate data on a three-dimensional sphere. But if you are working in GIS, then you are working on your laptop, which is two-dimensional. So you also need to translate the three-dimensional coordinate system into a two-dimensional coordinate system. And for that, you can make use of map projections as shown here on this slide. You have also different map projection models. Most famous one is, for example, the Mercator projection. Yes. And then if you then want to use data in GIS, then the data will always have a so-called projected coordinate reference system, which is basically a combination always of a three-dimensional geographic coordinate system and a map projection. And you have a lot of combination of them. They all have their unique identifier called the EPSG code. For Belgium, for example, you already have 3 commonly used CRS, projected coordinate reference systems that can be used. So it's always very important if you have spatial data also to know in which coordinate reference system it is. Okay, and then I will now give the word to Peter.
So good afternoon also from my side. I'm Peter Lottes, also working at ILVO. I'm responsible for high throughput field phenotyping at ILVO, and I will explain you more about rasters and how to get to traits and information about your experiments. First of all, remote sensing, the definition, it's the science of obtaining information about an object or a feature without physically coming in contact with that object or feature. Next slide, please. And for doing that, we use different types of sensors. So we can distinguish 2 types, passive sensors and active sensors. The passive sensors, they use actually the sun's energy, the different wavelengths that's coming from the sun and the reflection of it through the object, and we try to monitor that using an RGB camera, a multispectral camera, hyperspectral camera, or a thermal sensor. They don't emit their own energy or their own light. That's the reason why they are called passive sensors. The active sensors, on the other hand, they emit their own light or energy using longer wavelengths than used by the sun. The active sensors are, for example, radars or lidars, and they can also be used during day, but also during night. They can also see through clouds, for example, while with passive sensors, this is not possible. I will continue to talk about passive sensors here. Next slide, please. So if you are using passive sensors, we are thinking on related to RGB, red, green, and blue sensors. So the traditional camera, which is also on your smartphone, which is able to capture or see within 400 to 700 nanometer. And if we go to the multispectral or hyperspectral sensors, we are always also looking to the range of 700 to 1000 nanometer. There are also specific sensors for the sphere region, which is more related to the components within the plants, and that's up to 2500 nanometers. And then if we look to the thermal images, the thermal information which is relevant in combination with the draw trials, then we are checking on the wavelengths between 8 and 15 micrometers. Next slide, please. If we check then which light is available from the sun, and this is the left slide, this is shown in the left slide, then we see actually the yellow curve on top. This is the curve, the spectrum from an ideal black body at a temperature of 5,900 degrees Kelvin, which is the temperature of the sun. So that's the spectrum which is the sun is emitted by the sun. This is not the spectrum which is reaching the Earth's surface. That is actually the lower one, the black one with the visual colors between purple and red. So that is actually the spectrum which will be used by your passive sensors. You see that it is not anymore a continuous spectrum because there is absorption for specific bands by gas molecules like nitrogen, oxygen. You have large depressions due to water absorption. So that is the spectrum which is available as light that we can use with the passive sensors. And if we then look to the right-hand side, the top figure, there you see 4 different objects, being vegetation, being water, or soil. You see that actually the reflectance is clearly different over the different wavelengths. So those characteristics can be used to distinguish between vegetation and, for example, soil. But even within a vegetation, and that's the lower right curve, you see that there are different reflectances that they even for green vegetations, that they can differ. And also based on them, we can calculate vegetation indices, which use actually values of reflectance for a specific wavelength or combinations to actually distinguish between objects that are of interest. Next slide, please. So here you see two examples of sensors that we can use on a drone. On the left-hand side, we see an RGB camera, just an ordinary professional camera. On the right-hand side, you see a multispectral camera, the MicaSense, which is able to measure specific bands between 400 and 900 to 1000 nanometers. You see them also in the graph where they are really measuring. So on the upper two lines, not the continuous bar, but just above, you see actually the spectra which they are measuring or that we can measure using the MicaSense. But those are very similar to the bands that are also used by satellites. For example, the top row is actually the bands which are measured by Sentinel-2. So also there you get similar data. However, the resolution in which you get the data will be different. So the resolution for the RGB sensor is from millimeters. We can even measure sub-millimeter when needed, for example, for biodiversity monitoring, to centimeters, and the multispectral is around 1 to 2 centimeters information that you can get out. Next slide, please. Compared to, for example, Sentinel-2, which is a satellite often used, this information is free, so you can check on this, but this is more used to overview regions. It's less useful for following experimental plots because pixel resolution, depending on the band, is 10, 20, or 60 meters. Furthermore, as they are satellites, if clouds are between the satellites and your region of interest, you are not able to see what is happening in the field. So this satellite, Sentinel-2, they use the same bands, but it's more on regional level that you want to monitor, while with the drone, with the UAV, we have really high resolution and we can observe interesting traits in field experiments. Next slide, please. So in summary, we have different resolutions. We have the spatial resolution of a raster, can be very fine, very detailed with UAV data, or very coarse, but overviewing a region with satellite data. We have the spectral resolution. So how fine, how many bands, spectral bands can you distinguish? With a multispectral camera, it's limited to 5 to 10 bands for one sensor. Over hyperspectral data where you can measure every nanometer or every second nanometer. And then you also have your temporal resolution. If you decide, or if you are interested in dynamic traits, for example, you will fly every week, and then you have also a temporal resolution. So those are actually all the resolutions you have to deal with. Next slide, please. Now, how do we come from a UAV flight to image data? So we start at number one, top left. If we know which field we are going to fly, we need to make a flight plan. So we also know which sensor we are going to use and what is the field of view of that sensor. And then what we want to do, we want to have overlapping images, 80% in front and inside, because we want to actually stitch all these images to one big 3D model or a big puzzle. So there is software that is able to develop such a flight plan. So based on the amount of overlap, the size of the field, but also the resolution you want of the final products, a flight plan is made. This is loaded up to your drone, up to your UAV, and then the drone will automatically execute the flight plan, and that's what you see in item 3. Here you see the green lines, and those are the flight lines that are being flown by the drone. And during that flight, all or single images will be captured, and that's item 4 at the lower right. Based on those images, you will do a stitching using Metashape or Pix4D Mapper, but you can also use open source software such as WebODM. And the thing is that you will build a 2D and a 3D model, and you can clearly see this is a maze field, and you can see the positions of where the images are taken during the flight, and you can clearly see the 3D model, which is built upon those single images. Based on that 3D model, we will extract two products, being an orthomosaic, which is a big 2D image, but with a very high resolution, so can go up to millimeter resolution for the entire field plot, depending on the traits you want to determine. And what you also get out of it is a digital elevation model that is a height model, because you have each object in 9 different pictures, and you know where the pictures are taken in a XYZ location. You are able to extract really the height of the object. And what you see in the digital elevation model, it's a black and white image, and the values of the pixels are actually the height. Not only the height of the object, but it also includes the height of the field. So it needs to be normalized for the height of the field, and then you can access the canopy height of the crop which is in the field. Next slide, please. Based on that, so you have your bands or your RGB image. Based on the reflectances of your different spectral bands, you are able to calculate vegetation indices. And those vegetation indices, you have several. Here you see 2 examples, X is green and NDVI. Often they are normalized and they are sensitive to a specific trait. For example, X is green to greenness, and NDVI is often used to assess earliness and an emerging crop. But you have several related to chlorophyll indexes or to leaf area index, etc. Next slide, please. And then, if you have an entire plot, what you can do, you're interested in dynamic traits, and what you see here, it's a recent publication related to soybean, and what we wanted to do there is actually test the effect of inoculations of soybean. So what we did, and that's shown in the figure on the left-hand side, you see the time series. You see the green lines, those are the different phenological stages, and you see actually several drones. So actually, those were the time points at which we did drone flights. And what you see on the right-hand side, on top, you see the curve for 2 and the evolution, the dynamics for 2 vegetation indices for different plots. The one on the curve on the right is actually MS, stands for multispectral, and a vegetation index was calculated, chlorophyll index green, and you can clearly see the dynamics and the dynamics of the different treatments for inoculation. So the grain was not inoculated, and the other colors weren't different inoculations, let's say, and they really result in different nitrogen uptake and also different senescence. The second curve on the right is actually a SAVI vegetation index, which is more related to leaf area index. And you can also see that these inoculations have different effects. And below you also see that actually they also affect the duration of the growing season for different varieties. If you want to know more about it, please check the reference on the left-hand side below the slide. This only to give you an example what is possible. Next slide, please. So in summary, related to vegetation indices, I didn't talk yet about artificial intelligence of deep learning, but what are you able to extract? You are able to interpret static and dynamic traits. Static traits if you do one flight, for example, related to emergence or related to canopy closure for weed suppression. But you can also derive dynamic traits. How fast are crops growing or plants growing for a specific trait? So related to growth and phenology, but also for abiotic and biotic stress detection. But if you want to see growth dynamics over an entire region, then you should also check sentinel data, satellite data, and here are some links where you can find those kind of data. Okay, Arno.
Yes, so now I will end the presentation with a short introduction to artificial intelligence, machine learning, and deep learning. So as you can see here on the slide, AI or artificial intelligence, that's the overall general concept. And then you have more specific forms, which is, for example, machine learning, ML. And then you also have deep learning, which is an even more specific form of machine learning. So you need to interpret the figure as follows. Deep learning is always machine learning, for example, but machine learning is not always deep learning. But what's now the difference? If we are speaking about AI, then we are talking about all models for which you have to collect data, extract key information from this data, and then apply some defined rules to the data to make predictions. So it's really about mimicking the human brain in an artificial way. If you are looking at machine learning, then again, you need to collect data, extract key information from this data. But the difference now is that with machine learning, as the word says, the machine tried to learn the rules by itself for making predictions. So that's the big difference between machine learning and AI. And then you have the most specific form, which is deep learning. Here you always need to collect very large data sets. And the difference now with machine learning is that it also learns to extract features automatically. And then again, try to learn the rules for making predictions. So here in the training phase for a deep learning model, it also try to learn the most relevant features for making predictions. And why am I mentioning deep learning here? Because it is really good for computer vision and remote sensing applications. So you can do a lot with it for plant phenotyping, for example. How does such a deep learning model looks like? That's shown here on this slide. Basically, a deep learning model looks like the network you see here in the middle. So it consists of different nodes in different layers. You have an input layer, you have an output layer, and then you have also hidden layers. The hidden layers, there can be a lot of hidden layers. That's why it's also called deep learning, because you can have a very deep network. All the nodes are always connected with another node in another layer with a connection, and linked to this connection there is always a weight. And these weights represent then the features, the important relevant features that the model tries to extract for making predictions, or the rules for making predictions. So if you're talking about, for example, a classification model for plant phenotyping, then the model of deep learning works as follows. So you start with an image, like for example, in this case, an image of a rose. It needs first to be translated into numbers, of course, because you need to do calculations with the weights. So you give then these numbers of the raw image to the input layer. Then there will be a lot of calculations based on the weights between every connected node. And then you end with an output layer, which then can be translated into, for example, in this case, class probabilities, which then can be used for making predictions. So that's how a deep learning model works. It can be really complicated, but if you want to know more about deep learning networks, for example, you can always search on YouTube on tutorials. There are a lot of nice tutorials on this. How to train such a deep learning model. So if you want to train a deep learning model, you always need training data, and training data consists of training images. So you have images, and on the other hand, you also have labels, which basically indicates what's on the training images. And then you use this training set and give it to the model with starting from random weights. And then the model will try to make a prediction and compare it with the labels. And based on what the model predict and the label, the model then will try to learn and will try to adjust the weights in order to reduce the errors. And that's called then the training phase. And you do this for many images over many epochs, so over many iterations, so that the model keeps improving till you decide, okay, now the model is good enough to do a specific task, as in this case, classification. Okay, so that was a general introduction to deep learning and artificial intelligence. What's now the advantage of using also AI and deep learning, more specifically deep learning in plant phenotyping? Next to the things Peter just discussed, you can use deep learning in combination with remote sensing to do some interesting things. Like, for example, you can train models for doing anomaly detection, or train a model for biomass estimation, so that you just give an image to a deep learning model and that it then predicts biomass. You can also train models for counting flowers, fruits, ears, plants, etc. You can also do individual plant assessments, like, for example, in the article here in the right upper corner. This is an article publication published last year here from a colleague. And what they did here is they trained a model to identify individual plants by making segments around every plant. And in that way, they could extract diameters and heights per plant, also spectral values per plant, which then can be used for individual plant assessment. So if you want to check it, here is also the link to the article. And you can also use deep learning for example, doing weed detection also in combination with remote sensing. And I also added here on this final slide some interesting smartphone apps to do phenotyping with remote sensing or AI. The first one here is Canopio, which uses no AI, just kind of threshold exercise that they do to find canopy in a field. So you just take a picture then of the plants, and then it will try to extract the canopy of out of the image. But you also have more complex or more advanced apps, like, for example, Petiole Pro, which uses AI to, for example, segment leaves if you take a photo of the plant. So that was it for the introduction on the fundamental spectral spatial AI concepts for UAV imagery.
Thank you very much, both Arno and Peter. That was a really illustrative presentation with this introduction and concepts, and we are sure that we can have some questions from the audience during the last part of the session. So I would like to remind all our participants that we have enabled the Q&A section in Zoom for you to type your questions. So please don't hesitate to do so, and we will try to address them in the most appropriate time, and also during the Q&A slot that we have at the end of the webinar before we close the session. Thank you both very much, and now it's time to turn to one of the case studies today. In this case, the Lucerne case with Eric Roy and Bernadette Julier from INRAE. Thank you both very much for being there, and the floor is yours.
Thanks. So I try to make it better this time. Okay, so I will give the talk, but it has been prepared by Eric Roy, who is present today. And so we will be able to answer questions if needed. So this is an example, pixel from Phenotypes with a case of Lucerne. So it's first a description of the materials we are using. So the drone itself, we started with a first one, DJI Phantom 4 Advanced. I think this one break. After we bought a DJI Phantom 4 RTK until this year. And I think for this one, the battery is now over, so it's not possible to buy a new one. So we have a new DJI Matrix 4E. You can see the specificities of these drones. The workstation, we have also changed this year from, you see, from a Dell to a Lenovo, and with specific features. And we are also working with a NAS. So this is the material. The program, the method is photogrammetry. So here the workflow is not specific to our system. So we start with a set of images. We go to tie points. We produce point clouds, then DEM and the orthomosaic. So nothing specific here. And then we calculate the canopy 8 model here to represent the system for a plot. You have the green line is for the DEM, and you see the altitude of the pixels in the plot, and with some variation, of course. And you have all the pixels, and you have also the DEM just after mowing. We are on the forage crop. So this is a basis, or the basic line, I would say. And we have, for the calculation of the canopy 8 model, we use the altitude of the DEM minus the altitude of the DEM just after mowing. And the canopy 8 is calculated from a number of percentage of pixels that is the mean altitude of X% of the highest CHM pixels. So it can be 100% of the pixels, so all the pixels, or only 1% of the pixel, or whatever, between 0.1 to 100%. We have made experiments on 2 field trials. First one was during the BELIS project. We have, as an experimental design on CERN, a 4-block augmented design with 440 pure stand microplots with 387 varieties, and we had 8 biomass measurements with classical measurements by hand or machine. And you can see the design on the right-hand side with these microplots. And the flight plan, you have the characteristics here with an overlap of 80% as recommended, as said before. We have geo-referenced GCPs and all the characteristics. And the flight plan are represented with the red dot lines here. And okay. With this system, we have here represented the mean plot height measured either manually, so the red line, or by photogrammetry over 4 cuts in 3 years. And here you have the mean of all plots, so the 440 plots. So you can see, so you have 2 options for the imagery treatments, taking 1% of the highest pixels or 10% of the highest pixels. So first you can see that we obtain a relevant description of canopy 8 with photogrammetry. It's quite close to the manual measurements, but the option with 1% of the pixels gives a result that is close to the manual measurements. And so you can see the difference in canopy growth over the cuts and the years in this trial. And we have also used these results, this imagery, to try to predict the dry matter yield. And here you have a representation of the correlation between canopy 8 and dry matter yield. So axis, on the x-axis you have the canopy 8, and in y-axis you have the dry matter yield. On the left-hand side you have the manual measurements. So you have a classical positive correlation between canopy 8 and dry matter yield. On the top you have a representation of the correlation per cut, and on the bottom per year. Okay, so this is correlated, it's okay, it's normal. And on the right-hand side, you have the same correlation with the drone, the canopy 8 measured by the drone. And you can see again a nice correlation, and even better correlation between canopy 8 and dry matter yield. except for one cut here. And if you look at the plot on the bottom, you can see this blue points. So 1 point is for one plot. And here, in fact, in this cut, we have lodging, severe lodging. And in fact, in this case, the drone, the images taken by the drone, of course, is the natural height and not the stretched height. For all the other, when we have manual measurements, we measure stretched height, at least in this case. So here you can see that if ever you have large plots, you cannot predict dry matter yields. But in the other cases, the correlations were quite good. And here, important information that we have used, or Eric has used, 100% of the pixels to predict canopy height. So canopy height in this case is a good predictor of dry matter yields. Okay, so we have done a similar analysis with a new trial in a BELIS project for work package 4. And so the experiment has been shown in 2024. The experiment is more or less similar, but this time we have 4-block randomized design with 200 pure stand microplots and 50 varieties. So again, we have 8 and biomass measurements, and we have drone flights. And this time, so we have changed, we don't have the same, no. In fact, we have done some of the flight with both drones and some with the new one on. And we have, Eric has changed the flight length. So there are 2 missions now, the green one oriented east-west and the yellow mission oriented north-south. So what we can see now, here it's a comparison of manual measurements and drone measurements with 1% of the pixels. And here we have 4 cuts last year and 2 cuts in the beginning of the third regrowth this year. And you can see very, very similar canopy height measured by the 2 methods in 2025. Maybe a little under evaluation with drones for the third and fourth regrowth. And very, very similar in spring 2026. And here you can see the 2 curves for the 2 drones. So, so in fact, you can see very similar results with the 2 apparatus. So this is very good. So it means that you can change in the drone and it has no impact. It's yes, it is expected, but it's even better when you find that. As it's quite easy to have many measurements with drone flights, you can see here that we have obtained a full curve of regrowth. And you can see the lag time at regrowth in spring, and it's very interesting, and maybe to see also some differences in variety. We will see that later on. And you can also see in autumn that there is a stop of the growth because of onset of autumn dormancy in the sun. So it's very interesting to have this curve, the full dynamics of the curve. And here you have the correlation between manual height and UVA height, so quite a good correlation. And here's a representation for the 2 apparatus. And yeah. Okay, so there is a small bias that I think it's just that the drone flight, the drone measurements use the height just after moving, but the manual canopy use soil surface. Here, a view of genetic variation for canopy growth. So it's just a representation for 4 varieties. And in 2027, it's very interesting because, in fact, the red one The variety in red is a verdor, a very non-dormant variety that grows, that start growing quicker earlier in spring. And AAC Nikon is a Canadian variety with a strong autumn dormancy, so it starts later in spring. So, and the two other varieties are in between. So Karma is not very dormant, and Felicia is a Flemish type, moderately dormant. And they are completely well ranked in autumn. So autumn regrowth is just an indication of autumn dormancy onset, and so it's very striking to see the very good ranking of these varieties with these drone flights. In this year, we have also these curves of canopy age growth, but we have little variation among varieties. We don't know why, even if we have had very, very early onset of growing this year because of hot, mild temperatures in the end of winter. Okay, here are the figures that are of interest for all people who would like to start with drone flights and all this system. So the trials are not the same. The Euclid trial was a little bit larger than the BELIS trial, but with more plots, of course. We have changed the apparatus, but here you can see that we have while changing the drone apparatus, the time flight is becoming shorter and shorter, with 40 minutes only for the BELIS trial now with a new DJI M4E. We need a lot of memory, of course, so it means that we need servers. And with the new workstation, so not to make some advertisement for Lenovo, but because of its properties, the processing time is now of 6 hours instead of 16 before for the same trial. So everything is becoming so. Drone flight time is shorter, and the processing time is shorter too. So it means that if under you have many trials, you can process a lot of data. So as a conclusion, we have a good correlation between manual measurements and UAV-based predictions. We can share the protocol, and probably it can be useful for any forage legume in the stands at a vegetative stage, except if lodging, so it doesn't work if there is lodging. Of course, you obtain the canopy 8, but it's not representative of stretch plant 8 or biomass production. We have seen that if ever we change the material of the drone itself, it doesn't alter the results. So for the organization, you need material, of course. So a drone, a camera, a lot of memory, some know-how, and in particular piloting a license and the imagery processing. Our technician really like drone flights. It's much more comfortable than manual measurements of egg. And yes, everything is becoming quicker and quicker, and it's very good. So I have finished with this presentation. Thanks for attention.
Thank you so much, Bernadette, and thank you also, Eric, for this material and this presentation. Really interesting, and we have a couple of questions in the chat for this presentation and the previous one from Arno and Peter, and we will address them. These and all the rest to come in due time after the presentations in the last slot. So please don't hesitate to continue putting your questions on the Q&A section. And now we are moving to next slot, which is again with Arno, but this time also together with Michel Ney from our scope. And we are moving from Lucerne to Red Clover, and then we are dealing with flowers counting. So thank you both very much, and the floor is yours.
Thank you, Joachim. And welcome also from my side. My name is Michelle Ney, and I'm a forage breeder from Agroscope in Switzerland. And I'm giving this presentation together with Arno, who you have already met. And before we start, we would like to acknowledge all the people that were involved within this project. So from our colleagues from ILVO, Agroscope, DSV, and DLF that provided seed yield data, images, or models. And so in red clover, we have a lot of problems because the seed yields have been declining since decades. And one of the problems for sure is that the pollinator populations have been declining, especially the long-tongued bumblebees that are crucial for red clover pollination. But also the increased temperatures and the weather patterns are not very favorable for red clover anymore. However, these are all factors that we unfortunately cannot really influence. And in BELIS, it's all about breeding, so we need to find some traits that we as breeders can actually manipulate. And one of these traits is the flowering intensity. We have observed a lot of variation in flowering intensity, so accessions that produce less flowers, accessions that produce more flowers. And we believe that they have an impact on seed yield. And there is quite good indication from single plant level. So in diploid clover, the flower number as well as the fertility, so the seeds per flower, could explain 95% of the observed variance in seed yield. So the question we were asking within BELIS, can the variation in flowering intensity as well as fertility explain the seed yield in red clover? And to assess the flowering intensity well, we get the help of a drone. And this method, it will be the focus of our presentation today. So first, we went on to produce a very traditional color vision model that depends on color thresholding to detect flowers. So we used our orthomosaics and cut out 10 different pink flowers. Then we went on to look at the histograms of the pixels that were contained within these flowers. Specifically, we looked at the red-green ratios as well as the blue-green ratio. And what you can see here is that they are distributed kind of in a normal distribution. And we set some boundaries to determine which pixel values are likely flowers and which ones not. So all the pixels that had a red-green ratio larger than 1.1 and the blue-green ratio above 1 are likely flowers. And for the model, we also need some further assumptions. So we think that the flower sizes are between 15 to 1000 pixels. And for the flower to count as 2 flowers, we want to have a distance of 1 pixel or more between flowers. We put all these values in a blob detection model that is implemented in OpenCV in Python and let it run to detect flowers. And these are 2 examples how the images that we provided the model with looked like. So you can see green background and mostly pink flowers. And I have to mention, we also tried to do this with brown flowers, but unfortunately it was very difficult to get boundaries for brown flowers. So we just focused on the pink flowers. So if we let the model detect flowers, you can see with the blue circles, the detected flowers, it works actually quite well. So it doesn't detect the brown flowers, but the pink flowers are more or less all detected. However, if we use this model on different data from like orthomosaics from different breeding programs or that were taken in different lighting conditions or with different drones, we can see that it doesn't work very well anymore. So for example, here you can see some soil and it also detects flowers there. And here, when the lighting is quite different, it also doesn't work anymore very well. But with nice images with green background, lots of pink flowers, it actually works quite well. So because we were not satisfied with the quality of this model, we then went on to train an AI model. And Arno will explain you a little bit more about this.
Okay, so I will now explain which AI model we trained and the performance of the AI model. Yes, next slide, please. So we tried to learn a YOLO deep learning model for predicting flower heads on orthomosaics of red clover. YOLO deep learning model is basically an open source framework which you can use to train object detection models. An object detection model is a model that detects objects, and in this case we wanted it to detect flower heads in red clover. So you need a training data set, so examples of open and senescent flower heads, and also a test data set for evaluating the model performance. YOLO always predicts objects by making first bounding box predictions. So it tries to detect objects with bounding boxes and also predicts a class probability map. And then it combines the bounding box predictions with class probabilities to make final detection. And in this case, it was then for open flower heads and senescent brown flower heads on red clover UAV orthomosaics. For the training, so how did we train the model? We used a transfer learning approach, which means that we did not do one training, but we started with one training phase where we labeled a lot of flowers. Then we used the output of the first training phase to make prediction on new images. And then we corrected the predictions and used them as extra labels for a second training phase. And we did this for several times till the performance of the model was good enough. But of course, we had to start with a first label set, and for that we labeled a lot of flowers on different orthomosaics of different breeding programs, as you see here. So we used Label Studio to really draw rectangles around bounding boxes around flowers, and then we gave it to the model to then adjust and train the weights of the neural network for making predictions. We tried to select a training data set with a lot of variation because we wanted to have a model that has a big generalizability. So a model that also works on a diverse set of images. So here you can see the training data which was used. So you see a lot of variation. Also some temporal variation was included in the training data. And then we selected some data sets for training, which are here indicated with a blue rectangle, and then some for testing the model, which are indicated with an orange rectangle. The test data is only used for evaluate the model. So to use the model and make predictions, and on this test data set, the images, the model did not see these images before during the training phase. And we trained the model, of course, for the two classes now, so for the brown flower heads and also for the pink flowers. And here you see a result on a test image, for example. And as you can see, it's really good in detecting the pink open flower heads, but also the brown flower heads. We have at the moment now an F1 score which is higher than 0.95, which is quite good. The F1 score is a harmonized measurement, which tells something about the performance of the model. If it's 1, then this means that a lot of all the detections are really good, and also there are not a lot of flowers that are still missed by the model. In this case, it's really close to 1, so it means that we have a quite good model. What we also observed is that the model has a better performance for open flower heads, but only slightly better compared to the senescent flower heads. But that's also logic, because the contrast between the pink flower heads and the background is much larger than for the senescent brown flower heads. But still, also for the brown flowers, the model is quite good. Yes, next slide. And then if you, for example, want to apply then the model to an entire orthomosaic, as shown here on this slide, you can immediately calculate flower head densities per plots. So you can see here, and then you can see the variation in flowering density across the different plots, and then you can immediately find plots with high flowering zones and plots also with lower flower densities. If you zoom into specific plots, for example, you can also see the individual detections. If you zoom in even further, then you really see that it is able to capture also the very small flowers and also the brown ones. So really nice. Okay, next slide. And then the question is, of course, can we now use this model to say something about seed yields, or to say something about, can we use UAV methodology for predicting seed yield and relating flower head densities to seed yields? And for that, 2 trials were conducted in Switzerland from Agroscope, and one in Germany from DSV. So they had weekly drone flights during this flowering period. And they also measured seed yield and numbers of seeds per flower heads per plot, which we then could link to the flower heads predicted by the model. And we found that the pink flower numbers at week 5 and 7 before harvest were the best to predict seed yield in Switzerland and Germany. That's what we've observed from relating the seed yields to the flower head model. And you see also here, for example, here on the slide in the right bottom, that you see that for the pink, this is basically the density relative likelihood for the detected pink flowers, and you see that at week 6 in this case there are a lot of pink flowers, and then again afterwards it drops again to lower pink flower heads. Okay, and then if we look at the results, so if here at this plot we plotted the number of pink flower heads against seed yield for plots in Switzerland. And there you see that the correlation between the predicted open flower heads is quite good with the seed yield. And if you look at the correlation between the amount of the number of pink flower heads predicted multiplied with number of seed yield per flower head, then it's even better, as here on the right. And for DSV, we observed the same correlations. So again, if we related the open flower heads to seed yield, again, a good correlation with seed yield. And if we looked at the grams seeds per flower head multiplied with the predicted open flower heads, then the correlation was also very good. Okay, so what can we conclude now from this exercise? We can say, first of all, that the AI model outcompetes the traditional computer vision model for detecting flower heads, especially for the brown senescent flowers. Secondly, we can also say that drones can be used to reliably assess the flower intensities. It's more accurate, in the first place, more accurate than just give a score to the different plots by a breeder, because it's more objective if you use a standard model, and it's also, of course, less time. It's more time efficient. And we also observed that we had a quite good correlation between the predicted number of pink flower heads and seed yields. So this also shows that there is a lot of potential for using UAV methodologies for predicting, for example, seed yields. And the next thing, what we still have to do is now to explore if we also can use this UAV methodologies for selections in experiments. Yes, and this was our last slide. So thank you for listening.
Thank you very much, both Michel and Arno, and also the rest of the people contributing to this work. There are actually some of the BELIS partners were connected in the audience. So thank you everyone for For this, we are continue getting some questions and comments, and we will address after our next slot, which is as we usually do in these kind of webinars that we organize in BELIS. We always like to have some guest speaker from outside the consortium, and that's going to be today Corina Opliger from the ETH and Zurich. Thank you very much, Corina, for being there, and she will be presenting the case study this time on P. So, Corina, thank you very much, and the floor is yours.
I will start to try to share my screen. Okay, sure. Give me a second. Do you see the slides now?
Yes, perfect.
Okay, perfect. So that's then hello also from my side, and thank you for giving me the opportunity to present my work also in this round. I'm Corina, and I'm a doctoral student from the crop science group of ETH Zurich. And one part of my work has included to train a pea flower detection model, which is very close to what we have just heard, but yeah, I will do it as well, and it has a slightly different outcome. But I'm also an agronomist, so I do not only focus on detection models, so I'm showing you more also the practical application or what we have found. I would also like to start with a quick introduction of flowering, because flowering is a very important phase in a life of a plant, and it is the transition from the vegetative to the generative growth, and therefore also the start of yield building or yield formation. And also it is a very sensitive phase, and for example, too much heat or drought can lead to flower abortion and therefore yield losses. And for us, pea was the crop of interest since we have seen a rising demand from the food industry for pea protein, but I will get back to that a bit later. So basically, in classical field experiments, when evaluators do the rating in fields, the focus of flowering lays mostly on the determination on time points of flowering. So evaluators note the start, the main, and the end of flowering. But there is no quantification. Also, as we have heard in the red clover, it is also important to know like how many flowers are open in this case. And also from other work that what we have looked at when we just started with this project, then mainly flower detection was done with UAVs. And there we have seen that this pixel-based approach can, depending on the resolution that they have, the images, the accuracy can get lower depending on how high the flower density is. So others who have done flower detection, or for example in apples that have looked at single flowers, they were facing a different setup because apples, for example, are cultivated differently than arable crops. So we wanted to look at flowers in peas, which are a dense sown crop. And I have now learned from Arno and Michel that there are also similar approaches for what we did in flower detection, but with drones. But when we started back then, it was not so easy because I think the resolution was not so high in these drone images. So short about our research group, our core activity is to work with the FIP. The FIP is the phenotyping platform of ETH Zurich, and it is located in a field research station very close to the city of Zurich. And it contains these 4 poles you see in the image, and it is basically a rope-suspended camera system. You may know similar systems from football stadiums. So on the sensor head, in this case, there are currently 13 cameras mounted that allow to take images of plants from different angles and from about, let's say, 3 meters above canopy. And with that, we can take the RGB images very close from the canopy in high resolution. And for the approach that I'm showing today, we have used classical top-down images. And for the flower detection model, we have used over 280 FIP images, or patches of FIP images to train, and we have annotated them. In them, not only flowers, but also pods of 12 genotypes. We then trained a yellow 8 model. This is basically the same family that what has been presented before. And as you can see, for example, how we annotated the pea flowers with these bounding boxes. And we were very lucky because this is a visualization, so it worked quite well. And we also did some ground truth. We collected some ground truth data by counting the flowers manually in the field, and in addition, also on images. So as you see in these plots, on the y-axis is always the flowers that have been detected by the model. And on the x-axis, on the left plot, you see the amount of flowers that we have counted manually in the images. And on the right one, you see how many flowers we have collected manually in the field. We have seen that the flower detects the model detects these P flowers in the images with a very high accuracy. And also compared to the flowers in the field, there are still a lot detected, but some were not due to occlusion of other plant parts, but it was still very high. This is the wrong slide. Okay, we did also the same for pod detection. As I've said before, we also annotated pods, but with only one time point of ground truth data. That's why there are less time points. And as you can see, the detection of pods in images on the left side was quite high, but when we compared it to the ones in the field, it was significantly lower. And this is clear because the pods of the P are more in the canopy, while the flowers are more on top. So that's why we decided basically to go further with flowers. We did then more field years with over 180 genotypes in 2024 and 2025, and we did flower detection on FIP images, but also on literal images. I don't know if you're familiar with the literal. It is a handheld device that allows to take RGB images. And with these images, we were able to define start and end of flowering. And with that, we could also calculate flower duration. And also, we calculated the maximum flower density, which is basically the maximum numbers of flowers that were open in a day of a plot. And this resulted also in very high heritability. So between the genotypes, there were really differences between this maximum flower density. And we have also seen that it was correlated with yield. And that's why we wanted to see if there is even more that we can relate it to, and how could this be an interesting trait in further breeding approaches. And as I have said before, P is also a crop of interest for the food industry to be processed into meat analogs. And I have to give a quick background of that because mainly is of interest, P is mainly of interest in Switzerland. Compared to soybean, it does not have this GMO reputation. It is very easy to cultivate and it is non-allergenic. And it is grown for a long time now in Switzerland, but we do not know how well the varieties that we grow in Switzerland are for this food processing. And here protein solubility is a key trait for food processing. So we wanted to link this flowering, this maximal flower density that we have found to this protein solubility. So we tested 110 genotypes for protein solubility. And the results of these additional 2 years were very promising. We found correlations with yield, protein solubility, and yield with protein content yield and protein solubility with this maximum flower density. And to our surprise, the yield correlation and the protein correlation is probably more obvious, but the correlation with protein solubility is a very nice finding. And one possible explanation is here that the stronger reproductive sink, so more flowers may change the nitrogen allocation during seed filling, which may affect how the storage protein are accumulated. And this could have an effect on protein solubility. But I have to say here that this would need more further validation before it can be used as a proxy trait in breeding. But I think it is a very nice showing that like this high throughput field phenotyping traits that we could extract from these images can also have a higher impact even below yield. Also show a quick part on that. We have also conducted achievas for flower duration. I don't want to go too deep into that, but in achievas you basically look for differences, for small differences in the DNA that we call SNPs that could explain differences in the phenotype. And the dots you see above the red lines are basically SNPs that have been significantly associated with this trait, here flowering duration. We have not found one for maximum flower density, but here this for flowering duration. And I don't want to spend time on the biological meaning behind that, but I think this also shows that this high throughput field phenotyping that we use really is a nice complementary approach to GWAS, because for genotyping, it is also very crucial to have a vast amount of reliable and objective data to get meaningful findings. In addition, I would also like to show you this very pragmatic example. Here we have made more of like a thought experiment. We have also found a SNP for protein solubility that I did not show before. But this could be, for example, where is the difference if we would do like more a phenotypic selection in the upper panel or more a genotyping selection in the panel below. So if we took the genotypes that carry the SNP for the higher protein solubility, we would get the ones you see below. And if we took the genotypes based on their maximum flower density, you get the upper panel. For that, we took the 25% of genotypes with the highest maximum flower density. And generally, both showed similar results, but for protein solubility, which is a very interesting trait, we have selected, with the phenotypic selection, we have selected the ones with the highest protein solubility, which was not that clear for when we just selected it with the SNP. And also for the phenotypic selection, we have also selected for a bit higher yield. Also here, I would not suggest it immediately to a breeder to do that, but I think it is also a very nice example on how these traits that we can capture with high throughput field phenotyping can bring meaningful insights that we wouldn't even expect. And for sure, I would also like to give you a quick outlook. With the FIP, we are taking images from different angles, which allows us to make 3D reconstruction. I have not used it in this work at the moment, but in others. And this can be for the flower detection. It would be an interesting follow-up to make maybe also pot detection even more realistic. Also, it would be interesting to find out if we can track flowers and pots over time. But also, I should say here that this comes with limitations because we have trained the flower detection model with images that we have taken from above. And if we look at them from different angles, the results would probably not be that good, and it would need some retraining with different images. Yes. And as a quick summary, we have seen is that the pea flower detection was highly accurate. And also we have found these interesting relationships between maximum flower density, not only with yield, but also with protein quality, which may serve in future as a proxy trait. And I think also the fact that we have trained it on RGB images makes it more breeder friendly. And for me, it is also an interesting result to show that this high throughput field phenotyping traits really can serve as a complementary approach to genotyping, so that this goes together. And as I've said, in a future projects, I think the 3D reconstruction could show even more possibilities. And with that said, I would like to thank you for your attention. And if you're interested in other projects of our group, you are very welcome to check out the homepage.
Thank you so much, Corina, for this really interesting and really valuable contribution to this webinar. Now is the time for us to address the questions from the audience, and I would like to thank you all our speakers today for the time management. It was brilliant in order to have enough time for discussion now. And also I would like to thank Eric and Peter for these questions that you already addressed in the Q&A section, answering some of the comments. I'm going to try to address all the questions and comments that we have in order as they arrive. And actually, the first question was from SSG Lance, sorry about that, during the presentation from Arno and Peter and the introduction to the concepts. And this specific question is on how do I create validation data in field trials to model training, and how much training pictures is necessary to train a model, for example, maturity scoring or canopy height. I don't know if Peter or Arno, would you like to address that?
Perhaps I can start. If you're looking to maturity scoring or canopy height, I think for canopy height, the method is really well established. You can still check and do specific measurement of the height, where you also have an RTK GPS point where you really did your manual measurement. and then link that to the canopy height model, which is extracted from the UAV data. I think one of the other questions also related to that, related to the overlap, but also that the height is slightly lower. And I think this will always be slightly lower because actually the UAV model is never able to really detect the real fine details, and also as details or real fine leaves can move during the case of taking two images, it's very hard to really catch the top of the canopy. But what is more important, that you have the correct ranking, and that, for example, if you do time series, that you catch the dynamics. So in that sense, I don't think you make a big error. But no, of course, that's the exact measurement you cannot do. On the other hand, if you look to the ground truth and do all the measurements, canopy height measurements by yourself, by hand, I'm quite convinced that you will make way more errors than doing only one canopy height measurement using a drone in only 10 minutes. So in that sense, I'm still convinced that doing canopy height measurements using a drone is way more objective and precise. I hope this answers your question.
Yeah, I did an answer, but not I answered the Clemens instead of answering Akinto. But also, I think the one reason is that when we have plant measurements in a plot, we stretch the stems a little bit. So even if the plots are not lodged, maybe the stems are not completely erect. So there is this aspect. And also, I think there is just a human behavior that if we take 3 measurements per plot, so we put the stick somewhere in the plot, and we take the highest stems around the sticks. And so I think there is possibly a small bias with that. But I agree that I'm more confident now with the drone, the measurements made by drone after image analysis and with manual measurements. I think we can also check this with just analysis of variance, and probably the error is lower than with manual measurements.
Thank you. Thank you both. Yeah, we're gonna try to address all the comments as mentioned, and actually there was this comment about the percentage of overlap, which was already addressed by Eric and Peter, and I think that taking a look on both answers, it makes sense. Maybe it could seem a high percentage of overlapping, but after the presentations today and the discussions, I think that is clearly addressed. And the following question that we have, it was not a question, but it was a comment from F. Guerra in the chat. I'm gonna read it because it can be maybe addressed to. It was during the red clover flower counting presentation, but I think that can be addressed generally. This comment is: I often notice that depending on the orthomosaic generation settings, vegetation details such as flowers tend to appear or disappear to varying degrees. Am I the only one who has noticed this? Isn't it a problem? I don't know, any of the flower counting people would like to address that.
Maybe I can say something about it. So we haven't particularly observed that the flowers, only the flowers have disappeared, but we have observed that depending on the weather condition, it was more difficult or easier to construct the orthomosaics, and especially when it's windy, obviously the plants shift, and then it was quite difficult or even impossible to reconstruct the orthomosaic. And also, if on a cloudy day, it was easier to reconstruct them instead of when it's very sunny, you have all the shade and so on. So I think probably you should try to fly when there's no wind because that complicates orthomosaic creation. But we have not observed that specifically the flowers are missed in the orthomosaic creation. maybe someone else can add to this.
Yes, sometimes some flowers sometimes there was some distortion in the orthomosaic, but still we had no problems detecting the flowers, especially when they were pink. Then the contrast between the other pixels was high enough for detecting flowers. But yeah, for the rest, we had no problems with flowers disappearing. Yeah.
Thank you. Thank you. Anyone that would like to, I think that that was quite accurate enough to answer that. And any of the rest of you, don't hesitate to contribute with more comments if that's the case. I think that the question and comment from Arquinto was already addressed by Bernadette, not in the correct question, but it was actually addressed. So according to the order that we receive the questions, the next one will be the one from Clemens Langmeier, which was done during the presentation from Corina, but I think that this could be addressed in general to any of you. And the question is if have you ever tried counting male maize flowers? She mentioned that we gave it a go, but didn't get a good result. Any of you have experience on corn maize male flowers that could be used for that?
We got the question too once, but so far we didn't do any flights related to that. I also think, yeah, pragmatically, those are things that move a lot in the wind. So I think you should do high resolution imagery. So I'm thinking about millimeters because those are fine details. And if you want to catch them well and you want to do a flight on a non-windy day, that would be the best options in my opinion, as far as I can see. But we didn't test it either yet.
Good. Thank you. Thank you. Yes, there's another pending question. Well, actually, is based on several questions from Deepak Basavengouda. Sorry if I'm not pronouncing that correctly. I'm gonna just read the first one, and I would encourage you, our speakers today, to address this if it is on your interest to do so. The first question is, how does the methodology differentiate the red clover flowers from other pink flowering species that may co-occur in the survey plots? If other species with similar colored flowers are present, for example, brown canopy, how does the method prevent them from being misclassified as red clover? I think that's good, maybe for Arno or Michelle.
Yeah. We tried to train a model based on the most correct labels, and then sometimes you have indeed some other flowers present on an image, but we did see that there was not really a problem because the model didn't detect them as open flowers most of the cases. So we had not really problems with other flowers which looked the same as the pink flowers. So it's really there is a difference in how they look. So if you see it with your naked eye as well, then the YOLO model is also normally able to make a distinction between them.
A solution could, if it's really a problem for you, a solution could be to make it a separate class. Often a deep learning model, when trained well, it can differentiate between different type of flowers, and putting those extra flowers, if you have weeds in your plot in an extra class, that can solve the problem if it's trained also for that extra class.
Thank you. Thank you. Thank you, Peter. I forgot to say that we saw a couple of hands raised in the audience. I don't know if if this is related to any of the questions, but I would ask you to please put your questions in the Q&A section or in the chat, because this way we keep the mics for the speakers. So please don't hesitate to put your questions if you have some in the Q&A section or in the chat. With this question that we were addressing, as I mentioned, formed by several questions, there was another one that I'm gonna read again. Are photogrammetry derived outputs being used to detect and identify flowers? I assume this is the approach if the objective is to count total flower density to avoid potential overlap, since image matching, resampling, and orthomosaic generation can reduce or alter fine-scale visual information, has the potential loss of detail relative to the original imagery being assessed. That's a long question, but anyone would like to react on that?
Yeah, we now always trained on the orthomosaic just because then it's also, if you want to apply then also the model on the orthomosaic, you have immediately an overview of the predictions on the entire orthomosaic, but it could also be an option to train, for example, a model on separate images, because then you don't have the problems of the stitching, of course, and then you don't need, but of course, still, if you are not doing an ortho rectification of your photos, you still can have distortions also at the edges of your image. So this can still be a problem, but it might also work.
Good, thank you. Thank you, Arno. And the last question of this group of questions from this participant, thank you for that, is how are flowers that are not visible in the drone imagery accounted for example, flowers that are obscured by leaves, vegetation, or because of the viewing angle?
It only detects flowers which are visible, of course, because just flight and then nadir view, so we only see the flowers that are visual in the image or on the orthomosaic. So it's not accounted for the other ones.
Yeah, of course.
Maybe we did some manual counting of a part of our plot as well, and we saw that around 60% of the flowers were actually not detected. But one has to say that our plots were really voluminous, so yeah, a lot of flowers were like in the plot hidden. But still, if we do a correlation of the annotated images and the ones we counted, there is quite a big high correlation of 0.82. And that was with the classical computer vision model. So it's probably even higher if we correlate it to the AI-based flower counts. So I think the values are not entirely correct that we get from the image annotation, but the ranking is probably the same between the plots. So the ones that have more flower in real life or in the ground truth also have more flowers in the images.
Thank you, Michelle. Yeah, we still have like a couple of minutes more for more questions. So I would encourage everyone to raise some. Actually, I have one regarding the F1 metrics that you mentioned, Arno, and during the presentation of Arno and Michelle. But that is really nice, really high F1 over 0.95 up to 1. You mentioned that obviously when you work with models and you start getting these outputs, at the end, these F1 results, you keep on training the models in order to be improved and to get to good enough results that you expect. My question is how did you get to this over 0.95? I mean, what was the previous F1 score that you decided still to be improved?
Yeah, we did different training phases, more than 10, and we always calculated F1 score for every training phase. And then for the last training phase, we did not see any improvement anymore. So that's why we decided to stop training the model. because we think it's not possible to improve it even more. So that's why.
Thank you. Yeah, Corina, please go ahead.
Am I allowed to ask a question? Okay, cool. I have a follow-up question for Michelle. Do you know by any chance, because you started with the pollinators that should go to the flowers, right? Do you know if maybe the flowers that are visible by us are also more visible by the pollinators, and by that they are maybe the more important flowers to be detected anyway? I don't know, it's just a thought that they are even more contributing to seed yield in the end if they are more visible.
Yes, I had this thought before that probably they are on the outside of the plot and they are also accessible by the pollinators. So I think I would also agree with your statement that they are more important for seed yields than the ones that are somewhere in the canopy.
Okay. But you did not like check it. I don't know how you would do that, but
No,
you did
I mean,
advise.
you could probably by like collecting the flowers on the outside of the plot and the ones on the inside and then check whether the seed yield is different, but we have not done that.
Crazy work to do. Yeah. Okay.
Yeah, even without it, it was like a lot of work that
Okay, I
to
can imagine.
prepare
Yeah.
all of them. Yeah,
Thank you.
but maybe we will plan to do more of these trials, and I think I can maybe do a subset sample of this and then check. Good idea.
Yeah, it will be interesting for me, but I think it's a lot of work to really do it. But yeah.
We can do it in 10 plots or so.
Thank you. Thank you both. Yeah, I think we have this comment. Bernadette, would you like to raise
Yeah. Yes, it's a question for Corinna. I'm surprised by the correlation between flowering date or flowering duration and protein solubility. Was it something that was already known before? Because you don't need to have an estimation of flowering date with imagery to detect this type of correlation.
It was actually nothing that was known before. We're also like kind of surprised, and I'm also not sure why it has an influence. But my hypothesis would be that if the flower density or the flowering behaviors changes, so the zinc, basically the source zinc, the zinc changes, and then maybe the plant stores the protein different. And if you have a different ratio of legumine and vitellin, the protein solubility can change in the end. So maybe this, but it would need further validation. Also, you should test like the protein composition in the seed in the end if this hypothesis would be possible. But we don't really, I cannot explain exactly why it is that case, but we found it an interesting finding. Yeah.
Yeah, it's something like it's because you have these images that you were able to measure flower density. So it's a new trait, in fact. And
yeah, exactly. Yeah, and it was not that correlated with flowering duration because flowering duration would be like possible also with the manual ratings, and the protein solubility really was most correlated with the maximum flower density.
Yeah. Sorry, I mixed up everything, but but it's just because you have these images, you were able to measure new traits, and finally, it's correlated with another component that is important for P.
Yeah, it was also a bit of luck for sure to find something like that. Yeah.
Okay, thanks.
Thank you.
Thank you both for this interesting exchange. And we are getting to the end of this webinar. So first of all, I would like to thank all our lecturers today and also the participants online. We are 2 minutes before we end this session. So this is the end of this webinar. Image processing pixels to phenotypes to advanced legume breeding, organized in the framework of the Horizon Europe Project BELIS. So as I mentioned, we would like to thank all those who joined the session and all the BELIS partners who have presented the project, and also Corina as our guest speaker today. and we were lucky to see the progress of the project on this phenotyping on different crops, and the progress on image-based remote sensing and high resolution UAV imagery into quantitative phenotypic traits. So thank you all very much for your contribution and your attendance. And we are now closing the webinar. Thank you very much to everybody. Take care and see you next time.